Thank you for the opportunity to participate in this Great Wall ICC meeting. Yeah, this is my second time to join these meetings. Yeah, I've been to Beijing for many times, but it's very exciting, and thank you. So I was trained as a cardiosurgeon, although I'm not practicing anymore now, but we established our research team in Taiwan and the US, focusing on developing new strategies and try to prevent heart failure after injuries. So over the years, we have been examining the local and the systemic factors which can contribute to cardiac repair and regeneration after injuries. And we developed strategies for either improve the early protections, improve angiogenesis, or developing injectable hydrogel to control the mechanical properties of the heart, and develop the tissue engineering cardiac patch, try to determine the endogenous signaling pathways for improved endogenous cardiac repair. And we work on adult cardiomyocyte reprogramming and then cell therapy. So today I'm going to focus on the cell therapy part. So I'm going to focus on the use of human-induced pluripotent stem cells, or iPS cells. So just for those of you who might not be aware of this field, basically this is to use a gene transfer to overexpress 4 embryonic state genes, include OSKM, and then we can convert any type of somatic cells into pluripotent stem cells. And these cells can be used for many applications, including in precision medicines and cell therapy. So we create Taiwan's core facilities we call Taiwan iPSC consortiums since 2015. And this is a joint effort. I'm in charge of the facilities. So we generate iPS cells for researchers in Taiwan. And this far, we have serviced more than 40 institutes in Taiwan. And so far, we have generated more than 500 lines of human iPS cells, including in normal, control, and also across 45 disease iPS cell lines. And among them, more than 100 lines are successfully deposited in the public available Taiwan iPS cell bank. So if you are interested in any cell lines, you're welcome to check our website and contact us. And not all, but most of those lines can be delivered to you for research. And then we thought about how can we use these iPS cells for different applications. So one thing we are interested in is use this iPS cell-derived cardiac cells for drug toxicity and drug development. So I want to take these studies published in our colleagues at our institutes. So they report that the human leukocyte antigen HLA, the HLA-B 1502 haplotype is closely related with carbamazepine-induced Stevens-Johnson syndrome. So carbamazepine is a popular used clinical drug for convulsion. But it has been found that in certain percentage of the patients, if they receive this drug, then they might develop Stevens-Johnson syndrome, which is a disaster skin diseases. So we test if we can use population-based methods to generate HLA representative haplotypes, iPS cell lines, and then use that for large-scale drug toxicity screenings. So we screen about 1,000 volunteers in Taiwan, and because among the HLAs, HLA-B is the highest variant HLA subtype. So we sequenced the HLA-B first, and interesting. Although we only sequenced about 1,000 people, But comparing with a much larger database, this is a 2G bone marrow transplantation program, which it contains about 400,000 people. That's the largest bone marrow transplantation programs. So they have the database for HLA-B. So we find that among those HLA-B haplotypes, although we only have about 1,000 data, but the prevalence of those highest HLA-B haplotypes are identical to a bigger 2G database. So with that, we go further to sequence all the other HLAs. And then eventually we were able to identify 13 HLA homozygous donors. And this is just a list for those 13 lines. And we find that if you only check the HLA-B, then these 13 lines are able to cover about 52%, more than a half population in Taiwan. And because some of the lines of HLAs are overlapped, so we are able to select 3 lines of HLA homozygous donors, which are able to cover about 43% of HLA-B in Taiwan. So we went ahead to generate all the 13 lines of iPSLs and division them into cardiac and neural differentiation. So we generate 2D and 3D heart and brain organoids, and use that to do a high throughput, high content drug screenings. And we were able to test for more than 2,070 clinical compounds, and then test on the cardiac 2D and 3Ds. And then we use 3D cardiac organoids to test, to confirm and validate the cardiac toxicities. And we also do electrophysiology studies and even in vivo injection those potential cardiotoxic compounds in the mouse models. And so we are able to identify some previous unknown compounds with cardiac toxicities. For example, the second generation, the EGFR inhibitor, the Patonite. And similarly, we use the same platform and differentiate into a brain, the 2D and 3D. Again, we test their neurotoxicities, including in the brain organoids, electrophysiology, and injected into the mouse brain. And we were able to confirm their neurotoxicities of those compounds. And again, we identified several previously unknown neurotoxic compounds, including amiodarone. So with that, we argue if we can use these population-based HLA homozygous cells to develop cell therapy strategies. So we particularly focus on 3 HLAs, including the HLAs A, B, and DRB1. So which are the 3 major HLAs when we are doing clinical organ transplantations. So we wanted to test if we can develop those, can select those homozygous HLA cell lines, populate those cell lines, and develop as a super donor iPS cells. And interesting, we found that just if we examine those HLA, B, and DRB1, these 3 HLAs, and 2 lines of the homozygous iPS lines were able to cover 12.8% of the population in Taiwan. So meaning that if we generate a clinical grade iPS cells based on these 2 HLA haplotypes, then we're able to cover 12% of Taiwan's population, about 3 million people. That's a perfect match for the HLAs. And then we were able to find the HLA database from overall about half of the 8 billion population around the world. And these 2 lines can be used to cover 400 million population around the world. And as you can imagine, particularly here, that most of those people we can cover using these 2 lines are living in China, especially the southern part of China. But just in the United States, these 2 lines can still be used to cover 8 million population in the United States. So Then we've been working on how to use cardiac cells to do a cell therapy for the heart regenerations. So over the years, we have developed several models and then find that in the adult hearts, actually cardiomyocytes and endothelial cells, they can interact and crosstalk with each others. For example, from my previous work, we show that endothelial cells can secrete PDGF-B and binding to the PDGF-beta receptor in cardiomyocytes and existing surviving signaling for cardiomyocyte survival. But that's for mature cardiomyocytes. And then we started from embryonic stem cell derived cardiomyocytes in both mouse and human cells. We show that by a co-culture direct cell-to-cell contact through that manner, embryonic stem cell derived endothelial cells can improve the maturation of embryonic stem cell derived cardiomyocytes in terms of the sarcomere structure, mitochondrial structure, and also the electrophysiologies. So with that, we have generated 3 lines of human iPS cells, and we successfully differentiated each line of the iPS cells into cardiomyocytes and endothelial cells. So first, in cell cultures, we show that by co-culturing these human iPS cell-derived endothelial cells, we can improve the maturation of the human iPS cell-derived cardiomyocytes. Then we tested in the mouse models. So we performed the myocardial infarction models in mice and followed with injection of either vector alone or cardiomyocyte alone, endothelial cells alone, or combined with cardiomyocyte and endothelial cells. And another control is a twofold cell of cardiomyocytes. This is to provide another control for the cell numbers. And then we show that in all of the cell therapy groups, we can detect improvement of the cardiac functions. But the combined therapy with both endothelial cell and cardiomyocytes can achieve the best recovery of the cardiac function. And then we record a short period of time for the electrophysiology, and we find that the combined therapies, we can detect the fewest frequency of the cardiac arrhythmias. So with these results on hand, we also tested the infarct size and also the angiogenesis, because for the injection of the endothelial cells, we thought without the cardiomyocytes, we were able to find that the vascular densities is significantly increased by the therapies. And again, we found that the combined therapies can achieve the best benefits for improvement of the infarct size and also angiogenesis. And then we examine the graft size. That's a human graft in mice heart. And also the sarcomere length, indicator for maturations, and also vascular densities and the proliferation of the cardiomyocytes. In all of these parameters, we found that the combined therapies can achieve the best results for cardiac function, growth vessel, and then also maturation of the human cardiomyocytes. So with the collaborations at University of Wisconsin-Madison, we performed non-human primates models there. So first, we tested in the healthy monkeys, and we can inject either human iPS-derived cardiomyocytes alone or along with human iPS-derived endothelial cells. And we find that in all of the animals with the injection of human cells, and obviously we need to combine with the immunosuppression programs, and we can detect a large pieces of human myocardial tissues in the monkey's heart. And then in the combined cell therapies, we detect a much better, much more matured human myocardial tissues in the monkey's heart. We then move to ischemia reperfusion models. So following the 90 minutes of ischemias, we waited for a month, and then we performed the cell therapies in those monkeys with ischemia reperfusion injury. Then we use a cardiac echo, catheterization, and cardiac MRI to examine the cardiac functions. And And we found that the combined therapies, comparing with the cardiomyocyte therapy alone, we can improve, we can reduce the infarct size. And again, in all the animals, even under the ischemic reperfusion injuries, we can successfully engraft the human myocardium in the infected monkey's heart. And we even show that with the co-injection of the endothelial cells, then we can detect a direct connection, integration of the human iPS-derived cardiomyocytes with the monkey's cardiomyocytes. Yeah, we use remote monitors to examine the ventricular tachycardia episodes. So surprisingly, compared with the cardiomyocyte injection alone, both of the either injection of cardiomyocytes or together with endothelial cells, we can detect a significant increase of VT episodes within the first week after cell injections. But with the combined therapies, we find that the arrhythmia is quickly subside with the endothelial cell co-injections, suggesting that these endothelial co-transplantations can improve the maturation of the cardiomyocytes. So with that, currently we are working with a local company in Taiwan. It's a new startup. So we are developing a clinical grade super donor iPS cells. So hopefully we'll be able to move forward to a clinical trial in the near futures. So this is just named our collaboration, especially at UW Medicine, and also our long-term collaborator, Joe, at Stanford. And part of this fundings was supported by a new startup called River Biotech. Yeah, and I want to make a quick advertisements. So we created Taiwan's Circulation Research Society early this year, and we have a few friends here, including Chen, Yajing, Xinliang, Joe, Hishang, Jay, Tim Camp, Yibin, and Yu here. So we have put together a very exciting program for the 2025, and we are planning for the meeting for 2026. So hoping to see many of you will be able to visit Taiwan and give a talk at our meeting. Yeah, we'll stop here. Thank you.